So, to get this kicked off, so there were a lot of questions that have come through about how patients can find clinics and participate in studies and enroll in clinical trials, and definitely understand that there's a lot of enthusiasm there. I think the best way for people to find that out is to send an email to info@omf.ngo. OMF is tracking all of this information, and they will be very happy to provide whatever information they have, but they're definitely the best resource for that. That database is not in our speakers' minds. So there was also a really nice couple of suggestions on that people just want to help. They want to help us raise money. They want to help us raise awareness. I think this is really touching and really beautiful. We're so glad that people want to help do that. The answer is yes, please, please do anything to raise awareness. I mean, I think, we're in this community and we feel like everybody knows about this because we're sort of beating the drum over and over again. People don't know about this disease. It's starting to change. I mean, now I'm hearing, I mentioned unrest to some people and they'd heard about it sort of independently or watched it on Netflix, and that's really great. But the reality is it's still under the radar for most people. So Ron mentioned, talk to your congressional representatives. If you and any patients out there who want to help us out by making videos, making one-pagers, making documentaries, making anything, it is all more than welcome. So please do reach out, help raise awareness, help meet OMF's 2020 goal. Anything that you can do is enormously appreciated and helpful. So thank you for offering that. All right, so I think Ron's talk got us set up to some good categories of things that we want to discuss with this group. The first one I want to touch on is diagnosis. That's always been a huge challenge with this disease. It takes a really long time. So I'd like to talk with our speakers this afternoon about how what we've heard today can help with diagnosis. And definitely one of the aspects of what we heard that sparked some excitement were the wearables and a lot of the technology that Mike showed. So a couple of things. So Mike, do you want to first tell us what wearables you actually wear and what you like and what you think could be useful for the disease?
Well, I, of course, hello?
Yep.
Great. I like them all, of course, but smartwatches are they're going to be $20 in the future. They're $400 now, but they're going to drop pretty quickly. And they measure quite a few things. They're capable of measuring much more. And, I don't know for sure if I knew exactly what was going to work, would be happening already, but I think they're very simple to employ, and I'd be surprised if we can't find some things that are useful from this. And just listening, for me, this has been incredibly useful the last 4 days because I've heard from a lot of you folks today and also over the last few days about some ideas like blood oxygen drops. Could be, just for fun, we put it on a few people during the break, and their levels were actually the same as mine, at least at resting, but it wouldn't surprise me if your drop wasn't higher, say in slightly hypoxic conditions, meaning low oxygen. So I could see things that would be incredibly trivial to try that could be easily measured. If you think about most diseases or issues that are out there are actually easiest to find under perturbation. So stress echoes, right, finding heart defects is found when people run on a treadmill and other, I actually think airline flights are actually a stress for your lungs and probably for oxygen. So I think we could actually set up some very simple things that way that could be, pretty trivial that anyone could test. And in an ideal world, I love what Ron's doing and Juan and others, but actually in my ideal world, it'd be a simple home test that you guys can do to actually see what's going on, because then you can track for yourself what's working for you and what's not. So ultimately, we've got to get to that point where you can do this right off. And that's, to me, what's attractive about wearables. What am I wearing? I think I mentioned already the ring and the watches and the radiation monitor and the glucose monitor and exposome stuff. They're all good.
So we're going to come back to glucose a little bit later, but there was a question What second? And there was a question about SpO2 and HSCRP. Apparently, HSCRP has been measured as high in some ME/CFS patients, according to one of the submissions we got. Is that something that you could see incorporating into a wearable?
Well, it does go along with inflammation. Now, that will show up in a lot of other contexts too. Certainly, insulin-resistant folks, who are not necessarily ME/CFS folks, will have elevated CRP. It bounces around quite a bit. So it could be one possibility it's associated with some folks. Now, here's where maybe Ron and I differ. I'm actually a splitter, not a lumper. So, in its truest form, it is true that if you go all the way down, everybody will be their own case. Now, I'd like to find something a little more intermediate there. We probably agree. But I do think it's so heterogeneous. It reminds me a lot of diabetes, which is incredibly heterogeneous as well. So maybe there is a CRP form that's associated with high inflammation. That is quite plausible. And some of the cytokine results in Maureen are very consistent with that as well.
And why is it that heart rate goes up during illness? And again, anybody can feel free to jump in here, but that you saw your heart rate going up during illness.
Yeah, well, I don't want to hog all the airwaves here, but...
yeah, we're moving on after you, don't worry.
Okay, so... yeah, but, so, no, I think it's thought to be a natural response. You're actually elevating your temperature ultimately, right? It takes a little time, and so that's clearly one of the responses, and viruses don't replicate as well, nor bacteria in elevated temperature environments. So that's one way of it. Hope that wasn't a wearable there.
Wearable microphone. One way we
wearables are heavy.
It's all right. It's fine. Don't worry about it. One way we could combine the technology that Alain Moreau talked about with his squeeze test and wearables would be to have people do the squeeze test while wearing the wearables. And the great thing about that is that his little massage machine can go anywhere. And so this really could become your home stimulus that is what we lack right now. We need a little stimulus that doesn't cause you to go into terrible PEM, but that can produce a signal that's measurable. And if we've got 5, 10 different sensors on you from wearables, we're going to find such a signal if it exists. I think it's a good idea.
Just to add one thing, that you can actually make your SpO2 drop just by holding your breath. You can see it. So it's another simple perturbation.
And I want to bring Ron Tompkins' point earlier as well about the idea of the computer-assisted testing. I think that had a lot of parallels to what Mike was talking about with having your smartphone be your center and collect all this information that can then inform your doctor. How do you see all of that playing together?
I find it, particularly as we move forward, the patterns for medicine are going to be more team-related, and this notion of 15 minutes to get an evaluation is not going away. It is going to become more prevalent. So much of this disease is embedded in symptomatology, and frankly, there's not enough time in 15 minutes to really convey, and that frustrates not only the clinician, but certainly does the patient. And the extent to which you can get a good summary before you get there, that is so helpful for both parties. It's also good for communication among the care team. To have a reproducible, broadly accepted standard, I mean, one of the problems right now is that any particular clinician might have a favorite, and it makes it so difficult to compare studies. In the long run, we're going to have to do that. I can tell you, you don't go see a cardiologist without getting an EKG. You don't go to a urologist without getting a urine analysis. It's just basic. And they go from there. And in this disease, where all we know right at the moment, for the most part, are symptoms. And you have to have a reproducible, robust instrument in order to get on the same page in a very short time with your clinician. It also makes it simpler for the clinicians who don't know all the entire breadth of the symptomatology for ME. And lastly, it is going to be very useful to have a reproducible metric for if you're treating fatigue, There's lots of different types of fatigue, and you need a reproducible metric to know whether your intervention is actually doing anything. So I consider it, as a doctor, it's like if you have heart disease, you're going to get a cardiogram. If your kidneys are messed up, you're going to get a urine. And in this disease, from what we know right now, not having a biomarker, you got to get the symptoms straight.
Yeah, that's an excellent point.
I wonder if
Yeah.
reaction time would make a good, actually simple home test monitor you could do, right? That's something you can do reproducibly over
You could
time.
easily do
And
that.
it's all randomized tests
Yeah.
too, so that may not be a bad one to explore.
Exactly.
Yeah, and these are a lot of great opportunities, so it's really great to have Mike working with us. So speaking of devices, I think that leads nicely into the other talks that we heard about the devices that are being developed to look at patterns in the blood, the nanoneedle and the blood cell deformability. So I'm going to ask Ron and Juan now to, that rhymes, to talk about these two findings and how they might be reconciled with one another, and how that might actually, that's telling us about the physiology of the disease.
Well, for one thing, we don't monitor too much about the healthy controls. One of the issues, I think, is that what does the healthy controls do before they come in? We have seen cases where they had an appointment with us and they're late and they ride their bike across campus as fast as possible to get there, and they're totally out of breath when they get there. What effect does that have? And that may be true in a number of cases, that the healthy control went out running before they came in. And a lot of tests like you have to worry about, it's fasting tests, that you can't eat anything before you go in and so forth. We don't take that into consideration, partly because we haven't seen an effect of that. Now we're seeing something that we have to now start to consider. Why is there this heterogeneity that we are seeing? And so that gives us something new to try to explore. If that is the situation, we don't have to worry about the patients going out and riding their bicycle too fast. So they may be much more consistent because of that, and we haven't been worried that much about the healthy controls. And that's one of my takes away. We have to explore that a little bit. And then it's up to the engineers really to think through how they're doing that measurement and what that might mean. It's difficult because it goes from engineering to medicine. And the physicians don't understand the engineering, and the engineering don't understand the medicine really well. But so what we have to do is get together so we have kind of ability to try to figure that out. A nice thing about Stanford is that their engineering school is within walking distance of the medical school, and that isn't the case in many, many places.
That's you can't swing a dead cat without hitting a couple engineers.
Excellent visual.
We could try that anyway. So, I'd like to add to something Ron said. My background's in fluid mechanics, and we put particles in flows and measure velocities. And some of these are unsteady flows. Anyway, one very clear thing, this is technology for the last decades that have been refined. One very clear, important thing from that experience is the effect of sample bias. And I think it's worth thinking about for this disease, that not only is there heterogeneity among the healthy control, but I think by definition, the patients from which we would get samples are probably correlated to less severity. And if we're looking for a biomarker, we'd like to see the most severe, and those are probably the rarest and toughest samples to get. And if they're severe enough, I think there's a problem of consent preventing us from measuring. And I'm not saying there's a solution, but it would, all the data, for example, I've showed, and many of the work I've seen from my colleagues is independent of the severity. But the severity issue is tied to sample bias, and that's not the way, not the best way to look for differences.
I want to pick up on that point about severity. There was a question regarding the range, this huge range of the severity of the illness. What are the panelists' thoughts on why that might be the case, based on what we know so far?
I'll take a shot at that one. Yeah. Okay. You heard from all the talks today, there are two kinds of us. There are the people who are the splitters, him, for example, right? And the people who are the lumpers. And I think we need both of these groups. And so the answer to that question of whether the is dependent on which one of those you are. Right? So I think that there's an underlying cause, one underlying cause. I don't know that I'm anywhere near it right now, but there's so much homogeneity among ME/CFS patients that I think that's where we're going to find it. But what if Elaine is right, or what if Mike is right? And we have to subcategorize these groups. Even my friend on my left here thinks that we have to have cognizance of the heterogeneity in the patient population. And so that might be true. We don't know. And so what I find at this meeting has been really nice is that the lumpers are converging and the splitters are converging. That there's a kind of two groups moving together towards the same goal, and somehow they are converging within their own group. So I'm much more optimistic than I was at previous meetings. The other thing that the OMF has succeeded in doing with this series of meetings is that there's always been this patient community, and now we've got a scientific community. I mean, I know the people who are coming to these meetings well. I see them here, and now I see them at other meetings around the world. People are coming from Australia, people coming from Europe, people coming from, what, Canada?
Crazy.
Crazy. Okay? But I know these people now, and we're communicating better. Alain accused me of being a populist for his device. That I really think that his massage device is a very cool idea for exploring this disease, even though he's from Canada. Right? So we are friends now, and that means that we can say what we think. And so there's a lot of that going on now. It's been true all week at the scientific sessions, and makes me really optimistic.
To a great degree, I'm going to completely agree with Rob. I found the interactions this week particularly thought-provoking. My general view of the disease model, this disease model, I think is compatible with both lumping and splitting. Until we discover Rob's homogeneous continuing cause of the disease that we can attack directly, my concern at this point, if we're dealing with treatments that are directed at specific clusters of symptoms, then our therapeutic needs to be, the population we're studying must be enriched in those who are at risk of benefit, or otherwise we'll continue to have trials that are not showing benefit. That heterogeneity will destroy any possibilities with trials. But I am just as hopeful as Rob is that there's an underlying cause that is persistent with the disease for which we can develop targets that would correct it and just solve the whole thing. And in addition to that, maybe even prevent it for others going forward. So I have, in many ways, I'm a splitter for treatment at the moment, but I'm very hopeful in being lumped in the long run.
Mike.
Well, let me give a slightly different perspective, which will also explain why I'm a splitter, which is, well, let me back up and say I also think the meeting has been fantastic and I've learned a ton. Actually, a lot of it's from you guys. But the reason I'm a splitter is because when I look at complex disease in general, it does seem multifactorial. Even genetics is often not single genes. I can pick a case like autism where there are some single gene mutations, and it's true for diabetes. One single mutation will cause a disease. There's something called Rett syndrome, where if you have that mutation, you will have an autism-type phenotype. And then there are other cases where it's combinations of genes. And so even the underlying genetics has different complexities. Doesn't mean there isn't a genetic component, but even on top of that, there's an environmental component. So, and I think the same is going to be true here, and I see it for virtually all diseases, whether it's diabetes, whether it's autism, whether it's virtually any neurological disease is multifactorial these days. So that's why I do think it's combinatorial. It doesn't mean there can't be, much like metformin helps 80% of type 2 diabetics. That's great to have one drug that can handle 80% of the people. That's fantastic. But there's still 20% of the people left out. I'm actually one of those 20%. I actually have to take, by knowing the underlying mechanism for me, for my type 2 diabetes, it actually helped me figure out how to solve my disease. I'm actually rare. I'm insulin sensitive and I make insulin just fine, but I'm unusual in that I actually don't release insulin from my pancreas, right? But once I knew that, I knew what drug to take, and that works like a charm. So that's the power. If we can really understand this disease, what's going on mechanistically, I think we can cure it for people optimally. And I'm hopeful there'll be one drug that will help everyone, but it doesn't mean there will. I think that is very optimistic thinking, that it probably will take several drugs, and some will work better on some people and others on others. And so by understanding the mechanisms, and I feel we're at Nowheresville on that. I think we've got to figure that out. That's going to be incredibly valuable, and I hope the treatments then will match that, and we can, quite frankly, cure everyone. That's the ultimate goal.
Thanks, Mike. Actually, that does really touch on one of the other questions that was submitted, which is the idea of, is there going to be one treatment or sort of multiple? The lumpers and splitters are even among the patient population here. So this is a good discussion. Ron or Juan, do you guys want to add anything to that?
Well, I'm a lumper. But I understand the problem that if you're going to treat the disease, you're going to have to treat all the different problems that are there. They're not going to work, and I totally agree with Ron Tompkins. It's really important in a clinical trial that if we have a very heterogeneous population, and if the drug is actually treating one of the components of this, it'll be a failure, and that's a real problem, even though it would help a certain fraction. So if we do see differences, we have to figure out a way to test for them to know why they're in that particular group. And that's a bit of a daunting task, but it's something that will be necessary. To some extent, in terms of the splitter, there's a lot of, nobody in this room has the same genetics, even though identical twins don't really. And it's that genetic differences that can cause people to behave differently in their response. So it makes it, to some extent, it means that the number of types of disease is equal to the number of patients, because every patient will be different. So the hopefully is that we don't have to split it that far. Hopefully we can split it into a few groups that in fact then would be available for treatment. But because the disease is basically very similar, I think there's probably one underlying cause, and it may be hard to figure out what that is. But that's what we should always try to do, because if we can figure the underlying cause, we have a much better chance of curing it.
Juan, with the red blood cell deformability, you also saw quite a bit of heterogeneity in that. Does anything you found there point to, and any available drugs on the market point to anything that could potentially address that? What is your thinking on that right now? And others can weigh into it.
Sure. Something actually that Ron brought up is the fact that we're not controlling, how they fast or how they exercise. I'm talking about the healthy controls. That we were struck by the heterogeneity among the healthy, and there's pretty good hope that we can perhaps control some of that, or we hope that we can control some of that and make it more homogeneous. If that's the case, then that, and that heterogeneity accounts for that distribution. So if you saw the widths of my histograms of overall for healthy versus CFS, most of those widths are these heterogeneities. So if we can control for some of these, we can narrow the distributions, and then perhaps there is a difference with the same type of measurements and data that we have. And associated with splitters or lumpers, I think everybody's now confessed whether they're a splitter or lumper on this stage, except for me. And I like to tell my engineering students at Stanford that there's two kinds of students. One who can extrapolate from limited information.
Good. We've got a good
For
characterization.
leaving us hanging there.
So going back to just in the last few minutes we have, there are two other areas that were brought up as potential common features. Let's go for the lumping thing right now. So the metabolic trap was proposed as a potential way to lump all of these patients, the patients of this disease together. So many questions around this, so I'm very sorry I can't get to all of them. Rob is going to be here afterwards, so feel free to harass him openly during the reception, as I'm sure you will. But one that stuck out, what kind of events might happen in a patient's life or genetics or other factors that would cause this sort of jumping over into the trap? And could that be by any particular medicines or treatments that people are trying?
So this is a really common question. I've addressed it often on the patient forums. A lot of people have asked me this in my role as a kind of scientific representative to the patient forum. And so I've thought about it a lot. Anything that inhibits IDO1 for a period of time, say 2 weeks, would build up tryptophan and potentially cross that critical threshold. And so then you would be in the trap. What are things that might do that? Well, nitric oxide is a potent inhibitor of IDO1. So any of the theories that you might have heard from other scientists around here, like Laurel Crosby in the back of the room, where nitric oxide is elevated, would result in an inhibition of IDO1 and a buildup of tryptophan, perhaps more than the critical point. There are lots of other possibilities. And for example, I've asked on the forums, how many of you became ill with ME/CFS after overtraining as an athlete? There are a lot of those people. A good number of people were national class or even world-class athletes who fell into this trap while being extremely fit and healthy. And so you may know that in the 1930s, there were 30s? No, 40s maybe, I forget. That doesn't remember, somebody will remember. There was a tryptophan poisoning problem in the US. This was just at the, sorry?
And in Japan.
Okay, it started there, but in Los Angeles, for example, there was a huge group of people who took tryptophan as a supplement. It was big at that time. It's now probably bigger for athletes. And some of them came down with ME/CFS, or at least something that looked a lot like it. So there's probably a way to overcome the transport block from outside the cell to inside the cell, put too much tryptophan in the cell. I didn't mention this in my talk, but that transporter, LAT1, that moves tryptophan from outside to inside, is what we call kinetically asymmetric. That means you need only a low concentration of tryptophan to get in, but you need a high concentration of tryptophan to get out of the cell. And that asymmetry is supportive of the metabolic trap. It keeps the concentration inside the cell high, and it makes it very likely that the only way we're going to overcome the trap is by activating IDO1. So those are two ways that you can stimulate the trap or get it going.
Thank you, Rob. Ron.
I don't know an MECFS, but what I do know in physical injury, of the stress of physical injury, the rate of, for example, all the amino acid transporters is grossly up. What's that?
Use the mic.
It,
Keep it up.
Just
keep it up.
Keep it up. Yeah.
In blunt injury, in all the tissues that we're aware of, and the genomics of all the amino acid transporters is dramatically upregulated. And in this particular, they just are, because there are huge increases in amino acid fluxes throughout the body. Now, what I do not know if LAT1 within the brain is included within that. I can easily imagine that it is, and I also could easily imagine a ligand that could be measured by PET, but it hasn't come up as an issue. But if you have a nonspecific enhancement in that asymmetric kinetic transporter, then you're going to get more tryptophan inside the cell despite any sort of specificity. So it's I would not be surprised if that's just simply a result of stress.
Rob, there was one question for Ron. Sorry, there was one question for you as well regarding heart preload failure. You had mentioned that as an avenue that you're looking to explore, and the potential tie to POTS mechanisms. And can you say a little bit more about that and how that may potentially be treated if that does prove, in fact, to be playing a role? Yeah.
Thank you for the question. I tried to explain this on the website, and I'm trying to get it in a language that is understandable. And so please check the website, and I realize it's a work in progress. I even have some doctors that I end up arguing with about this. It is, so this is not a cardiac nor pulmonary reason for the fact that you do not generate sufficient cardiac output at the maximum exertion. And in this particular case, there are two forms. There's a form in which the amount of venous return to the heart is insufficient to create the elevated cardiac output. And in that case, one possibility, in healthy people at peak exercise, there's a little bit of your blood volume which is shifted from the venous to the arterial side, and that provides more blood at the end of the heart filling to help you increase your cardiac output. There is some consideration for a generally low blood volume, particularly in women with MECFS, and that may play a role. We don't think it does because they're giving a liter of saline just before the test, and this condition still persists. And that's the low flow form of this preload failure. There is also a high flow phenotype to this, which is related potentially to an autonomic control or a peripheral uptake of oxygen. Let's just generally call it oxygen delivery. Both of those are a little bit too complicated for me to describe at the moment, but just as Michael was talking about, I believe, the arterial concentration in the venous side is a little bit more elevated often in patients, so the oxygen delivery may well be inadequate. And what is seen in that consistently is the mixed venous oxygen content. That's the oxygen content in the blood that has made it back to the heart is unusually elevated. That means insufficient amounts of oxygen were actually taken up within the peripheral tissues, and which is expected because it's consistent with the fatigue, the malaise, and the plateau that you can accomplish with exertion. And so they're sorting through the many ways that can be created pathophysiologically, I think is going to be a tremendous asset. And we certainly plan to be on top of that.
Okay, that's excellent. I did mention I would get back to glucose. We are running short on time, but I do want to mention, at least to Mike, that a lot of comments came in about impairments in glucose metabolism that have been observed in ME/CFS. So it seems like with the glucotypes and all of the stuff you were looking at, that this would be something worth continuing to pursue. So just to close things out, I want to invite each of our panelists to say quite briefly what they've seen this week that makes them hopeful. Juan, I'm going to start with you. No dead cats, please.
I'm excited about these controls for health control to try to reduce this. Also, we've just started looking at some of this capillaroscopy, and if we consider it pretty high risk, but also potentially very high payoff, because this would be, in vivo measurements. They're non-invasive. You put your finger under this microscope, and I think we could do much better than the commercial devices. So. And such observations would take into account many things, some of which have been said now about microcirculation and differences. And I think with computational power that we have now, we can analyze a lot of data.
Great. Ron?
I'm optimistic by what happened in the 3-day research conference, and that seeing all of the people there interact a lot better. We're all friendly. We kid each other a lot, but there's a change of saying, what can we do? And it's not just about, I've measured this and here's what I got. It's okay, what does that mean? And there's a bit of a change in that attitude, and I don't see that in other scientific conferences. And I think that's what has to be done. So it's really a unique opportunity. And I don't see it in the other ME/CFS conferences I've been to. And a lot of this is getting really, really smart people, non-arrogant people that can really try to think this through. This is a hard problem. This is a very hard problem. But I think the mindset is to be of one. And it doesn't necessarily mean that the patient community is left out. They make a lot of suggestions, and I very much welcome them. And they send me things, and Rob gets inundated with suggestions, and it is very important. So I'm optimistic that this is very much a big team effort to solve this very complex problem. And I think we'll continue to make progress on it. And what I'm hoping for is a breakthrough, and that's what you often see in this kind of an effort. People really working hard, trying to focus on the problem, and they do get a breakthrough. And the issue is when you're just collecting data and you're not thinking about the consequences, the breakthrough may be right in front of you and you don't even see it. And it's really important to be able to see it. And this is the right attitude to have, because your mind is focused on that. And when it comes up, as opposed to something like unexpected, that's weird, you will say, oh my gosh. And I've seen that in other fields as well. And I've seen cases where a discovery should have been made years and years ago, but was ignored by the scientists because they weren't thinking in terms of utility. They were thinking about collecting data. And we rediscovered it, and it should have been discovered 5 years earlier. So that's what makes me very optimistic. I see that change in this community, and that's great.
Mike.
I guess I've been most impressed by you, the community. I think it's been incredible seeing your energy to solve this, your fact that you aren't taking no for an answer. You want to see this get solved. I really think we can solve it. I'm a big believer big data will tell us the answer. I think we don't know what's going on, and let the data tell us what's going on, and that's why, and having you guys all participate, keep pushing us is a good thing. I just think it's incredible. So it was even showed up at the meeting. There were, as you know, patients there and advocates, and it was just great to see how eager they were. They were probably the most vocal people in the whole scientific meeting, were the folks who actually had the disease. So for me, it was incredible.
Rob.
Well, so as we get towards the end of the line here, all the words have been said, and so
That's okay. We're running out of time.
yeah.
I'll be really, really quick. I would like to.
No, you can still say something. I didn't mean you shouldn't say anything, please.
You didn't show the 5 minute sign.
2 seconds.
No, both of you can still say something. I mean, please. Yeah.
Okay.
Okay.
All right, so once again, we've exposed the splitters and the lumpers. What I love about the wearables and the big data is that many things get measured in time in individual patients. And, you know, I'm a kineticist. If time is on the horizontal axis of a graph, I love it. And that's what Mike can do. And we combine that with Elaine's massage or some other small stimulus, we're going to learn a lot, because now we can get cause and effect, right? The whole point of a time course is there aren't too many things we know about cause and effect, but one thing we know is the cause comes before the effect.
That's a safe statement.
And if all you're doing is measuring one point and then going to some other person and measuring that one point, you don't get any information about cause and effect. And so these little time course experiments are the ones where we'll find the cause.
Cause and effect, that's a breakthrough, Ron. All right, Ron, last word.
Okay. I would echo my friend Ron's comment about the team building of the science team. And Stanford is way ahead of us. We're just now coming online. But I so much enjoyed the week interaction, those with the disease participating and making suggestions, and the scientists all very much working together, and the quality of the information that was being exchanged at Stanford. I'm very encouraged, because I find that there's a tremendous interest to do exactly that same thing within our center. And I am very hopeful that we make great progress and in getting to this same level with that organization, and we're fully committed to work in a collaborative manner. I do have to say that resources, as you've mentioned multiple times today, is more of the limiting step in this process than it is ideas and ability to accomplish a lot. And so I have to say I am very encouraged, and I have great confidence that this will make some serious progress.
Great. Well, thank you. Thank you all.